Hey, welcome to HTM Hackers Hangout. It's June 7th. I've got a group of people here waiting, and so I'll get to everyone and we'll take questions. I know some people are watching. Hi, Hyung-Sung Go. I saw you on chat and YouTube, and hello, Brev. Good morning, everybody. We've got Marty online, and Paul, and Marcus, and David, who's been helping me with my Twitch stream and doing the React stuff. So I have a short agenda. First, I want to show off. So that's the first thing that I want to do. So let me show off real quick. And this is just because I want to show off the work I've been doing on Twitch, which is building HTM systems. This is, if you look down below, the guy on the left there is David Truman. He's been helping a lot with this. And if you want to see it actually working, you can go to this URL. I'll put it in chat and YouTube if anybody's watching. You can actually see the live app here. And this is the latest thing that we created just a couple of days ago, was this potential pools visualization. So we're going to keep working on this, and eventually we're going to add pros all around this and code samples and all that, and really fill it out so it looks more like a real interactive document, sort of like this. So we haven't styled any of this at all. I just want to point out, anybody who's missed what I've been doing on Twitch, this is basically it. And the code base is wide open. I've been working with the community, had quite a few contributions, not just from David, but from a couple other people too. So I'm showing that off. The other thing that I want to show is that we have had a lot of new projects, HTM projects over the past year or two that, I mean, I haven't really thrown up much fanfare for these, but if you look at this projects page, which again, I just threw up into chat, there's a ton of HTM projects in here, and I just updated this this morning. So some of these are really new. I want to point out a couple of them. This one, e-tailer, which Marty, who's the pink teacup in the forum and right now currently in chat, he has been working on this. So there's a lot of details. It's a C++ implementation, very, very fast. I haven't tested this, but I'm going to take his word on it because he's made other HTMs that were also extremely fast. Tiny HTM, for example. Check that out if you want to run parallel C++ HTM. That's a lot of potential there. Another one that I haven't said much about was there's one in Scheme. And I think this has recently been updated. It didn't have, yeah, so there's recent activity on this. So someone's programming an HTM in Scheme. So that could be interesting if anybody wants to get into that. I think Scheme is a type of Lisp, isn't it? Right? Correct me if I'm wrong there. And one more thing I wanted to point out was the Snowflake Medium article by Jimmy, which I really liked. We just had a Bay Area meetup, thanks to James, who's not joined us right now, but he typically will pop into HTM Hackers Hangouts. He did a great article about AI and what it is today, and how HTM is different, and why he put it into a plugin for his database called Snowflake. So he took Paul Lamb's HTM.js implementation and basically injected it as a plugin for Snowflake, the database. Oh, one more thing. Sorry, I prematurely turned that off. I also want to show, okay, so nupic.cpp. DMAC just posted this morning, so I wanted if anybody was working on Nupic CPP to have a chance to buzz in here. Here's a current sort of status of it right now. They're updating algorithms interface, making things run faster, but they are not planning on doing an OPF. That's fine. I never expected it to be an OPF in nupic.cpp. If there's going to be something like an OPF, I expect it to be in another repo like nupic.py, perhaps. But there is a network API, I believe, in nupic.cpp that should eventually, I don't know if it has PyBind interface to it yet, but I hope that it will eventually have a PyBind interface to the network API. Open question, if anybody knows the answer to that, let me know. All right. Right, so next up on our agenda, I talked about updates and some of the new HTM projects. Before we go to Marcus and do a little QA on research, anyone in the community at the moment want to highlight any of their work that I didn't touch upon, or have any comments on what I just went over? Crickets are fine too. Okay, let's go and do some Q&A with Marcus. I see there's one question on the forum thread for the Hacker Hangout that Falco said. He said he probably wouldn't be able to tune in, but so let me read his question here. Actually, let me just show it. Okay, so if I understand correctly, the reason why systems like grid cells and displacement cells are necessary in the neocortex is to cope with movement while receiving sensorial, let's say, sensor information. The fluctuating data due to movements needs to be compensated for, otherwise it would seem like a stream of completely new and unrelated information. When a cloud passes or light switched on, colors change, the brain still recognizes everything. Wouldn't a similar system like grid cells be required to compensate for changes in color? This is something Falco's brought up on Twitch before with me, and I think that his argument is like, why isn't color treated the same way as orientation? So in one of our latest Twitch streams, hello, Mark Brown, I just noticed you were on YouTube chat. Thanks for joining. On the way to the airport, I think you're headed to Jordan, right? Whew. See you in a while. Anyway, I digress. So there's this idea of color, and another question regarding another interesting comment that Marcus made. According to a paper that was discussed, a probe in a subject like a rat in an environment Marcus described as a jungle gym, which is like a three-dimensional sort of something that a rat can go in all 3 dimensions easily, produced a deformed grid pattern. Is it possible that this has the effect of a wraparound grid, as if the pattern was printed on a piece of paper and they wrapped around a 3D environment? Is it possible that the absolute distances between vertices remain constant, but that seen from the top of the projection of the pattern is what causes the distortion? So, Marcus, take it away.
Okay, yeah, sure. There were a few things there. I can talk for a little while based on those because there are kind of 3 things there. So grid cells and displacement cells. So to predict the input, to predict your sensory input, you need some context. You need something that represents context of some sort. And sometimes the right context you need is a location. For example, if you know the location of your retina in the reference frame of the object you're sensing, and you know its orientation, you know which way it's pointed, that is enough to figure out what the eye is going to sense. So in general, like in some ways I'm stating something obvious on purpose to build up from it. In general, to predict what you're going to sense next, you need some sort of context. Sometimes that is going to be a location. Sometimes it's going to be like what just happened, what few things just happened that's going to cause me to predict what's next. So you use the example of the passing cloud, for example. Well, that's like a movement sequence of the cloud moving. I could make a connection there to grid cells in the sense that as a cloud moves across your visual stream, the location of your retina relative to the cloud, the location of your retina in the space of the cloud is being moved. It is moving because of the cloud's movement, which is like, so if you do have grid cells tracking that, there's something kind of nice there that it will that you can use that as a clue to figure out what movement is occurring, or the nicer extreme. The nicer version is if all the clouds are moving, you can use that as a clue to figure out your own movement, your own self-motion. That's kind of like a form of optic flow. That's kind of the least interesting thing I'm going to say. So I think you made a good, you pointed out something good when you talk about color being similar to orientation, and you bring up displacement cells. I think the right way to think of this, I might draw a little bit on the board. The right way to think about what we often call displacement cells is I'll actually draw a diagram from I've been reading some Jeff Hinton recently, and a diagram from 1981, which is kind of similar to things I've drawn before, similar to some stuff in our papers. So let's see. Can you hear me right now?
Sounds good.
Cool. So this is notation from multiple papers from Jeff Fenton in 1981. I could point to a specific one, but the idea that you're going to transform between like what I'm sensing. I guess I would say like what's out there, what's actually there. And some sort of like a relationship or like I'll say transform or relationship.
Is that what's actually there box, like reality and everything I may not be able to perceive as well? Or is it just to compare what I'm sensing to?
It could be interpreted as that, but like this, for example, can be this can be as simple as like, here's what's landing on my retina, like here's an oriented edge or something like that. Here is that edge in the reference frame of the thing I'm sensing. Or so you know, I said what's actually there, but at the core, what we're doing is applying some kind of transformation. So like, didn't think through this exactly. I'm trying to write this in very general terms, but this is some sort of transformed version of this. Like it's like a normalized. I'll say it's like a normalized version of this sort of where like the normalized input, such that like if you were looking at a coffee cup, you were looking at the top of it, you can see it from lots of different viewing angles. This changes completely as you're seeing the coffee cup from different viewing angles, but this one's going to be the same if you're looking at it the same part of the cup.
So in our terms, we call it it's our allocentric reference frame of the cup or something.
Yeah, that's an example of what this can be.
Okay.
So when we talk about displacement cells, we're usually referring to where is one part of an object in the reference frame of the whole. Like for example, where is the handle in the reference frame of the cup, or in the framework paper, the Numenta logo, where is it in the space of the cup? And this idea of having some sort of, for example, we'll use that specific example. In the frameworks paper, we have a logo and a cup. The transformation being applied there is given a location in the space of the cup. Imagine if you're not familiar, imagine a coffee mug with a Numenta logo on it. Given a location in the reference frame of the cup, what is that location in the reference frame of the logo? By tracking this sort of like the displacement between those, the coordinate transformation between those, you can. This is the thing you want to remember about the cup. That's sort of where this comes into it. And once you remember that, you can now apply this transformation to arbitrary locations in the space of the cup, which means if you know where you are in one reference frame, you can predict what you're going to sense because you remembered this. Now, all of this is a way of saying, like, what I'm building up to here is there's another form of transformation that can be applied to all your input, and that is what is the if you think of the lighting in the room changing, then you get this sense of like what I'm sensing like, I don't know, my retina is getting hit by some mix of photons falling within some range. And then like what's actually out there, what color do I actually perceive that's out there? And I think it's correct to say that like the similar diagram, you're trying to infer what is the mapping from this to this, and you want to track this. You want to keep track of this at the current moment. You want to keep track of like, this greenish thing actually maps to blue, or like I'm inferring that the lighting in this room is such and such, therefore the dress is black and blue or whatever.
Wait. So let me try and infer what you're trying to say. So you're saying this transform may not always be just applied to location?
Yeah.
So, okay, so that could, so that's why you may be able to perceive different objects in different contexts where they look completely different and recognize them as the same thing.
Yes, I think yes. For like a second there you cut out, or it might have been me, I lost you, but yeah, like, yes, the idea that multiple inputs will be kind of normalized to the same thing based on some form of transform.
So I argued against Falco and this idea of color being somewhat represented in this way. So it sounds like that he might be on something.
I would say that the high level concepts of keeping track of the transformation between input and what's actually there, I can draw the analogy there. However, I don't know, representing the color of something using grid cells, I don't see anything there at the moment. It doesn't stand out to me, but I can step back and think in terms of the higher concepts of what's here and show that this connection exists.
I think I'm following you. I just never thought about it that way. So that's good explanation.
Let's see. So that was part two. I guess the third was totally disconnected. Let's see. Have I addressed this first part very fully?
Well, you've certainly provided some food for thought for me, hopefully for him too. Well, he'll probably discuss it with me on my stream later.
I might think of something later, but yeah, we'll see what happens. Okay. His third part, or his second part, was about an experiment that happened in Kate Jeffrey's lab, and the paper isn't published for that yet. She's basically they've just presented it in talks and such. I don't even know if there's been a poster on it, but there are multiple labs right now who are trying to figure out how do grid cells behave in 3D, or what are their firing properties as an animal moves in 3D. And Kate Jeffrey's group is focusing on rodents like rats and mice, and then Olanovsky's lab, I think he's in Israel, is focusing on, I think it's Israel. He's focusing on bats, bats flying around in 3D and tracking what grid cells are doing. None of these results are published. There have been a couple posters from Olanovsky's group. And so Falco asked about what I in a research meeting called the jungle gym. This rat is moving through this. We'll just keep calling it that. This 3D triangular lattice. It might not have been triangular, but the point is this lattice where the rat can climb around almost arbitrary directions in 3D.
Some type of isometric grid that it can go all over.
Yeah, and importantly, in these rats, first they experimented in 2D environments, found grid cells. Like they made sure to find a grid cell that is grid-like in 2D, and then they had the rat climb in 3D to figure out how those cells are firing. And it seems that the fields are these separate 3D blobs that are spread through space in a non-uniform way. It didn't form this clean 3D grid, and it didn't form something that in a paper that I did with Mirko Klukas and Ila Feet that's still kind of in progress, but we have a preprint out. We pointed out that grid cells could uniquely represent locations in 3D using multiple modules, even if the modules themselves are 2D, such that the firing field of a grid cell is more like this pillar coming out of the ground, like diagonally or straight up. And we pointed out that this could work. Which means these results made us uncomfortable because they are not doing that, as far as we can tell. And So okay, that's some background. So it's really hard. I would draw something on the board, but it's really hard to draw something that's 3D and messy and make it worth drawing. But so in that, Falco asked if well, first I'll throw in. He asked if are the fields kind of uniformly separated, or are the distances between them similar? So last I knew from seeing talks from both Kate Jeffrey and Nakum Alanovsky, basically Kate Jeffrey's conclusion, or that their group's conclusion, I think she's working with Roddy Greaves on that, was, and it still, it hasn't been published yet, so I mean they're still working on this, is that the fields are blobby and non-uniform, and that was basically where they left it. Whereas Alanovsky's lab with the bats, they also confirmed, or they also reached the conclusion the fields are like these 3D blobs. They're not uniform like a lattice. However, the space between them is roughly the same. The spacing of them is, I mean, uniform in the sense that like just the distances between the fields, although the directions between them might be kind of messy, the distances between them are roughly uniform. So it does seem like that's plausible that the fields themselves are kind of uniformly spaced. So I do think that Falco asked if what could be going on here is that this 2D grid is kind of being folded into 3D space in some funky way. Totally plausible. Yeah, that could be exactly what's happening. It might be the case that like as the rat moves around, it's always almost always attending to a 2D plane. So it almost perceives itself like, all right, now I'm in this 2D plane. Now I'm in this one. And it does that in sort of a way that, that I don't know, it follows a certain set of patterns. Like it always comes back to the same few planes.
Well, it's entirely possible that all these different organisms might have different mechanisms by which their grid cells represent space, depending on how they navigate through space, right?
Yeah.
So like the bats could be totally different from the rats.
One interesting thing here was that the studies, because they're, especially Kate Jeffrey's lab has done a fair number of studies of various types of 3D, whether it's like the rat is climbing pegs on a wall, the rat is climbing chicken wire, running through the spiral staircase. And one thing I think that they found through talking to them a little bit is that it does make a difference whether the rat kind of grew up climbing walls. If they didn't, then you expect, I think they found, none of this is conclusive. All of this is just like individual experiments. It's easy to overinterpret it, but early results with grid cells showed kind of these pillar-like fields, like when the rat is climbing these pegboard walls, then a single grid cell would often have a field that just kind of stretches up into space. But I think those were rats that were less experienced with a 3D world, where if early during development, if they just kind of live in 3D, if they navigate 3D all the time, it almost seems like maybe their grid cells learn 3D space differently. So it's kind of based on experience.
That's super interesting.
Sounds almost like a Riemann surface, because the field would be on rather than a regular two-dimensional plane.
So let's see if anybody has questions for Marcus while you got him. He's yours for the next probably 20 minutes at least.
And there's not really a need for me to give that much of a update or research summary, because like a couple nights ago we had a meetup here. So I mean, go on Matt's YouTube feed to see talks from mini talks from Matt and Jeff and Subutai, where they kind of summarize what's going on.
Yeah, it's sort of unnecessary to give research updates here in this meeting anymore, since we're just live streaming research.
Yeah. At the same time, like if you just jump into a research meeting, you're kind of being thrown into the middle of things. So yeah, the meetup gave some good stuff. Yeah, I mean, a couple of us are working on the high level question of how does the neocortex learn models of objects or represent models of objects? An object here can be like it's kind of a loose term. It's, if anything, it's how does the neocortex learn the structure of the world, with a particular focus on movement, with a particular focus on your own self-motion, and how does the brain learn the structure of the world via that. And then when we say the structure of the world, we kind of think of that as compositional objects. Structure, the world's structure is kind of arranged. I'll say hierarchically. I'm not talking about the hierarchy of the cortex though. The world itself is components arranged into objects, which are themselves arranged into objects, and which are arranged into scenes, which are in places, and places are their own bigger objects, and just neuromechanisms underlying how all of that can be sort of encoded and learned. Is a couple of us are working on that, and then the others of us are working on applying what we've learned to machine learning, to deep learning.
So there's a question from Hyung-Sung Go on YouTube. He says, how are they certain the grid cells didn't re-anchor during the observation? Talking about the 3D rat jungle gym experiment.
I would say they're not certain of that. That's very plausible that they are, that a lot of re-anchoring is occurring as the rat is like, it might get tired, stops for a second, and then kind of reorients, and then starts moving around again. And in those times, maybe the grid cells firing fields changed completely, because they're just doing these heat maps of grid cells over time. As long as their sample size isn't too large, as long as they're not recording the rat for like days and days and days, you're still going to find these grid fields. So that's very plausible. I think there's a really good chance that there's a lot of varying over time. Like pretty much these grid cells and place cell studies are very biased toward detecting things that can be discovered in a heat map. If you have this heat map of, you have this trial, you have the 5-minute trial, rat running around a box. You make a heat map of that, of all the places where a cell fired, and you look at that, and anything you can conclude from that picture, they're going to discover. But anything that gets lost by that picture, there's a good chance that those things are harder to find. It's just kind of the experimental paradigm.
What are the types of things that are hard to find that you wouldn't find in a heat map like that?
Like one thing is, what was the paper? Here I can point to exceptions where they did succeed in finding it. In the recent, who was it? There were recently 2 papers that came out. One was from the Stanford people, Kia Hardcastle, Will Butler, and the other was from, can't think of their name right now, that involves grid cells and rewards and learning reward locations rapidly. I'll post this link somewhere. And one interesting thing they found was that the activity and the entorhinal cortex and our hippocampus, over time, if they break it up into time slices of say like 50 milliseconds, I don't remember the exact time slice size, they found that the activity was kind of alternating between how the cells used to fire and how they are. So imagine rats in a room. There are rewards spread around the room in these consistent places. But then an hour later, those places are rearranged. Now the rewards are placed in different locations. They found that as the rat is running around this room now, it is sort of jumping back and forth between representing the room as if the rewards were in their previous locations and as if they're in the new ones. Basically, if you try to decode where the rat thinks the rewards are, I'm oversimplifying a little bit, but if you try to decode from the neural activity where the rat thinks the rewards are, the activity is like, it kind of goes back and forth. First it thinks it's in the old places, now it thinks it's in the new ones, now in the old, now in the new. There's this temporal juggling of like representing different possibilities that what this comes down to is the grid cells are firing where they used to fire, and now they're firing in their new location. Now they're firing where they used to fire, now they're firing in their new location.
Is this, could you compare this to like an intersection of two concepts?
Intersection. You say intersection, but I think you mean union or like.
Yeah, I guess you're right. Yeah, union.
It's both. Yeah, it's both of them. Yes, and so it seemed there was some sort of the heat map would, if you were looking at this as a heat map, you would see like, oh, this cell fires in its old location and in its new location. But if you look at it spread out over time, you would say like, but it never does both. It's like the cortex is jumping between modes, old mode and new mode.
So you could miss a lot of oscillatory effects by looking at heat maps over too long a period of time.
Yeah. Yeah, and there, you could say something similar could be going on in 3D, where it's actually jumping between modes of some sort.
That's interesting.
While this feed is going on, I'm going to find that paper. I feel bad for forgetting the authors' names.
Okay, while he's finding it, and still open for questions, you guys, trying to read some of the chat if there's anything. Mark Brown mentions, like, there's some optical illusions that seem uncomfortable, and he's saying like a Necker cube is one of those Escher-looking cubes, because he's saying it because it triggers repeated grid formation. You know, you're trying to navigate through an object, and you're every time you go from one point to another, it doesn't make sense, and you like maybe you're jumping to another grid formation or something.
Just to throw in that other paper I mentioned is called "The Entorhinal Cognitive Map is Attracted to Goals," with the first author being Charlotte Bocara. But anyway, that was a cool study.
All right. Well, I think we're going to wrap up. Marcus, I really appreciate you joining to take some Q&A. Oh, somebody's, go ahead.
Yeah, I have a question.
Yep.
Yeah, so currently the temporal memory layer has a problem of the repeating input problem. Is that a feature of the neocortex, or it's a bug in the HTM algorithm?
I would say I would say that there's not an equivalent problem in the neocortex. What Marty's referring to is in general, you mentioned the repeating input problem. In general, the temporal memory has this strange thing with any sort of shared subsequence, where if suppose A, B, C, D, E, F, G appears in multiple sequences, like sometimes it occurs in multiple contexts, the temporal memory will basically have to relearn it in all of its context, and that learning process takes a while because it has to kind of first it has to learn XA, then XAB, then XABC, and then it has to learn YA, then YAB, then YABC. And the repeating element problem is the special case of this, where the subsequence length is 1 or 2 or whatever you want to call it. So I would say that the temporal memory is the class that is called the temporal memory. It's sort of in its academic form of just being this pure algorithm that has these quirks like that. And we also have this older thing called we later changed its name to the backtracking TM. And that was more of a productized version of it that has that solves some of these issues through using things that aren't very biological, like backtracking. Backtracking is this idea of, before you burst, jump back in time a few time steps and say, suppose the sequence had started right here, would I have burst? And if the answer is no, it kind of jumps back in time, changes which cells are active as if the sequence had started it at a different point and continues forward. So that's I didn't try to explain that slowly and clearly. I was mostly just illustrating the fact that, or making the point that these issues, back when we had this productized version, which still sort of exists in the form of Grok, we came up with these like one class of solutions to these problems back then. And we can keep just making up answers to how this could be solved, how this system could be kind of biased toward trying to reuse previous sequences rather than thinking A, then AA, then AAA, then AAAA are all separate sequences that all have varying lengths. But at this point, we'd just be making stuff up at this point. So yeah, the repeating inputs problem is something that we think will settle eventually. It's certainly not a problem that the actual cortex has, as far as we can tell.
So how would a real neocortex distinguish between a subsequence and a real sequence of different lengths of subsequences?
At this point, I'm just speculating. I would guess that if seeing something as a subsequence or as a new subsequence succeeds in improving the neocortex's ability to predict its input, if something like suppose the neocortex juggles a few hypotheses. One of them is going to be like, okay, this sequence is something familiar. It's not new. It's not a novel sequence. It's the ABC that I'm used to. Don't say it's a different ABC. It juggles that hypothesis with, wait a second, this might not just be ABC. This might be XABC. This might be a different sequence. And it doesn't know which of those is the correct hypothesis. So it kind of tries both. And if it learns over time, if statistically it works out over time that this sequence, that interpreting something as XABC, if going back in time a few time steps and including that context, if that helps it predict better, then it will learn that subsequence. Then it will learn that, like, no, the X matters. The big question here is, like, the neocortex is wondering, like, does the thing before the A matter, or should I just ignore it? And I would say that probably it solves this by trying to both encode that previous element and excluding it, and whichever one causes the better predictions wins out over time. That's a high level answer of how I think that these things would be solved.
That's kind of contrived too, right? Because this is like a single stream with a single input on a computer rather than, massive parallel streams of sensory input. Campus kind of driving attention span and stuff like that could also play a deciding factor to make the decision on which one weighs more.
Mm-hmm.
Like, you can't tell the difference between 0.333333 up to 50 digits versus up to 60 digits. Your brain's not going to, those are going to be the same number, even though they're not the same number in your head. Or you look at a random pattern, you can't figure out any patterns from it because it's random. To me, that's kind of the same thing. Right?
Yeah.
Cool. Thanks.
Yeah, hopefully that was useful. There was just some speculation in there and trying to give you a little context on the fact that, yeah, there's this problem in the temporal memory because it's sort of been pulled back to its like academic form. It's not in its practical form. It had, like, if you want to apply the temporal memory in production systems right now, you need to add a couple things to it, like better cleanup of connections that don't prove valuable, or these kinds of issues. And those issues are there.
Because my current implementation actually just simply connects a new cell to every previously possibly active cells to avoid this problem. And
Okay.
the thing you have said somewhat proves my implementation.
I see, yeah.
Cool.
Very good. Okay, we're running out of time because we have a meeting at 10. So anything else, last minute comments or questions?
I had a quick comment on the repeating inputs problem, because that's kind of an area I'm interested in. A couple of things. One is, so if you think of a sequence as an object should have semantics, so there should be semantically similar sequences. So if you think about, a cortical column sort of learning a more structured thing, a more complex thing as it sees it more often, then you could see the subsequence becoming features, themselves a feature of multiple sequences. And the other idea is that timing could be involved. So, if instead of saying I'm doing L, 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 instead you say I'm doing L for X amount of time, then, that's a different way of sort of encoding the information without having to worry about L versus LL versus LLL.
I totally agree with both of those. And the somehow having this, I don't know the right terminology, but for this, but some notion of grouping the input stream into these pieces, like A, B, C, D, E, F, G, is like, we don't think of that as this strange sequence of an A followed by a B. We think of it as like, no, it's part of the alphabet. Whereas other subsequences are totally random to us. So how's that kind of grouping occur when you get just the straight input sequence? Something like that is probably going to be needed, but I'm just speculating at this point. And yeah, the timing thing is a good point too.
All right, anything else? Thanks everybody for joining out there on YouTube and joining live. I think we have 8 viewers right now. So appreciate everyone joining in. Don't forget to watch me on Twitch later today. We will have a research meeting in about half an hour. And I'll also be building HTM systems after that's over with. So thanks, Marcus, for joining us. I really appreciate you helping out.
Yeah, it's fun.
All right, take care, everybody.